This is a video to recap cellular differentiation in meristems and stem cells. Okay, so what is cell differentiation? This is a process by which a cell expresses certain genes to produce proteins characteristic for that type of cell. And that is really beneficial because it allows us to have a huge variety of different cells that carry out specific functions depending on what proteins they make. So if we look at stem cells, and these are regions of unspecialised cells in animals, and unspecialised means they don't yet have a function, they haven't differentiated yet. And in stem cells, we say that all of their genes can become switched on. And when we switch on a gene, it means that they start to make those proteins. So if we looked at a red blood cell, for example, when they differentiate, they would switch on a gene that codes for hemoglobin, and that is going to allow them to carry oxygen around the body. Whereas they might switch off a gene that's maybe associated with something like cell division, because they don't need to be able to do that. And again, with all of these different types of cells, they're going to have different genes switched on depending on what their function is. The other process that stem cells can do is they can carry out something called self-renewal. So we can see that this stem cell has divided, and that just allows them to make more copies of themselves, to make more stem cells. So in plants, we call these unspecialised cells something different. We call them meristems. And meristems are exact same as animal stem cells in that they can self-renew. So they can divide to make more copies of themselves or make more meristems. And they can also differentiate. So remember, that is just them expressing certain genes to produce proteins characteristic for that type of cell. And that's going to allow our meristems to differentiate into lots of different plant cell types. You'll remember things like phloem cells from Nat5 and your root hair cells. So looking at the two types of animal stem cell in a little bit more detail, we have embryonic stem cells and we have tissue stem cells. Embryonic stem cells we find in the early embryo, and that is sometimes called the blastocyst. And we say that they are pluripotent. And pluripotent means that they can differentiate into any type of cell. And the reason that they can do this is because they can switch on all of their genes, and we'll talk about that more in a second. Tissue stem cells are found in specific locations in the body. That could be things like bone marrow, or it could be in the skin. We say that they are multipotent, and multipotent just means that they can differentiate into all the cell types in a particular tissue. We'll look at an example of that in a second. And they're involved in growth, repair, and renewal. And in terms of growth, that just means allowing the tissues to increase in size. So think like when you're going from a child to an adult, we need our cells to grow. And repair allows us to replace new cells in things like cuts and allow the tissue to heal. And renewal is when certain tissues are constantly losing cells. Think about like the skin. They continuously have to make more cells in order to function. Okay, so just looking at the difference between multipotent and pluripotent. When we think of our embryonic stem cells, we said that they were pluripotent. And that meant that they could differentiate into any type of cell. So they can switch on all of their genes and they can make proteins characteristic to any type of cell by differentiation. And if we were to look at our tissue stem cells, they have a much narrower range of things that they can differentiate into. We said that they were multipotent. They can only differentiate into cells of a particular type of tissue. So when we think of our blood stem cells that we find within our bone marrow, they already have some genes switched on. So they'll have genes associated with blood switched on already, and that means that they've got a narrower range of things that they can differentiate into. They could become red blood cells or white blood cells or platelets, for example. So looking at the two types of uses of stem cells, we have therapeutic uses and research uses. And broadly, I think of therapeutic uses as helping sick people, and research uses more to do with studying the disease itself. So therapeutic uses involve the repair of damaged or diseased organs or tissues. So we know that embryonic stem cells can self-renew under the right conditions within a lab. So as a therapeutic use, if we had someone that maybe they were going blind, for example, we could take some corneal stem cells and spray them onto the damaged cornea, and this would cause more corneal cells to be produced. Could also be really good for someone that maybe has really bad burns on their body. Previously, you would have had to have used skin grafts, but if we could take some skin stem cells and spray them onto the damaged area, that would cause more skin cells to be produced naturally by the body. So research uses involve stem cells being used as model cells, and we can study how a disease develops. And that is really good as it means that humans don't have to be tested on. We can study the stem cells in a lab in a controlled environment before we test them on a human. And it also provides us with information on cell growth, so that's just how cells grow and divide. It can give us information about differentiation, so how they become specialised, and it can also give us some information about gene regulation. So we know that every cell in the body has the same DNA, but not all the genes are going to be active in every cell, so we could get some information about gene regulation from stem cells. So how could we actually use stem cells to repair damaged tissue? Thinking about muscles, for example, we know that they weaken as we age, they can become damaged in things like accidents. How could we replace that muscle? Let's say it was in someone's leg, for example. So what we would do is we would first of all culture those stem cells. So that just means that we allow them to divide in lab conditions. And then once they've divided, we would transplant those stem cells onto the damaged area. And regardless of the particular tissue that you're asked about in your question, you're going to use those words that are highlighted in pink there. You need to say that you're culturing the stem cells. It could be corneal stem cells, for example, and then you are transplanting them onto the damaged area. Okay, and lastly, we need to be able to briefly summarise the debate about stem cells. A lot of people disagree with using them as it can involve the destruction of an embryo. This is because a lot of research involves the use of embryonic stem cells, which have more potential for differentiation due to them being pluripotent. However, it can also provide effective treatments for disease and injury due to being used as a model cell in research. So we need to be able to give the pro and con argument here.